Okay, let us go back on to the lecture part. This is also getting in our Breeze account. So for your understanding, you can also go ahead and get the URL site. Okay, the same thing, http breeze.uhv.edu/histology1 PowerPoint. Okay, you can take down the HTTP site so that you can get this PowerPoint printed, and then you can study and write your homework and everything from that point. Okay, but eventually you will get it onto your computer. breeze.uhv.edu/histology1 ppt/. Okay. Here it is. Just gone through all of them, but we will have some figures too. Okay, here it is. Here we go. Don't want to get in. Okay, this is the topic title, histology. Okay, it's running fast. I don't know. This is the introduction part. Just like an objective which I gave it to you in the class on the familiarize yourself with the components of cell. Okay. How many of you have done in your earlier class about the of the cell? If you say immediately in your mind, you should like a rectangular one. If I say a cell, right, and then a round nucleus from maybe 6th grade onwards, or now you get a 3rd grade or 4th grade science, right? The basically science, right? So imagine you are like a kid of 6th grader. Okay, now just for today, and we will go into step by step. This, what is this? You are going to teach me now. Yes, right? Membrane, plasma membrane, plasma membrane. Okay, this one is nucleus. Okay, so what is this one? Cytoplasm. Cytoplasm. Okay. So this is an animal cell or a plant cell? Plant cell? Yes. It's a square. That's why you said in the plant cells. No, it's not like that. But plant cells, you will get a cell wall built on surrounding. So here you have cell wall. So when you see the cell wall there, then that's on the plant cell. But without the cell wall, and if you see the cell with the plasma membrane, and then nucleus, and then cytoplasm. So generally, we are talking about here in this class is the animal cells on histology. Okay, we are going to do that one. So in my situation in this class is to explain to you first about the cell architecture, and then if you have too many cells, it forms just one cell, okay, and then this is a one cell, 2, 3, 4, 5, 6, 7, 8, 9, 10, like that, okay. So you have several cells, and this is, we call it as a tissue. Follow? So if you have one cell, we call it as a cell biology, as well as we call it cytology. Cytology deals with the individual cell, okay, and tissue will indicate the collection of same type of cell. This is a one type of cell, okay. So the same cell on multiple. So this is a tissue, a kind of tissue. So one kind of tissue. You are going to deal with different types of tissues in this class. So first we will study about the cell, one, and then we are going to study on the tissue, and then we are going to study collection of tissues. Collection of different tissues that is constitute an organ. Follow that? So first we will study the cell. Okay. Okay. Now it is better. Cell, and then tissues and collection of different tissues. Tissue 1 or tissue 2, different type of tissues together form the organ. Right. The cell, hey, you know about plasma membrane, cytoplasm, and nucleus. That's the cell. That's fine. That's a cell general outlook. But there are different types of individual cells are there, right? Blood cells, plasma cells, and then heart cell. That's an individual cells, right? As well as if you look into the microscope on the blood, there are white blood cells. There are different kinds of white blood cells are there. So they're individual cells. They may vary, okay? And the cytology is all about I think in MD Anderson, they used to concentrate on the cytotechnology. Just study the cell type. They are giving 1 year, I think, in a diploma or certificate, so that you can go for a job. They won't give you a job. It's right when you go and walk in, hey, I want to have a job. They want you to get another training for cytologist, a cytotechnologist. Then you are able to get a job over there. So for that, they are giving 1 year training on cytology. Hey, I know what is cell and what is cytology. That is not enough. You have to go and study what you are going to study, how to get the individual cells from the bloodstream, and then you are going to stain them up, and then you are going to look under the microscope, and then the proper treatment of the cells or the plasma or the blood, and then to study the individual cells. So that's the part that you are going to do it in cytotechnology. So in histology, in this class, we are going to study more about the tissues. That's why histo means tissue. Histo. Okay. So whatever the biology of the tissues, that's called as a histology. And biology of cell, cytology or cell biology. So that's an entirely different class. I think sometimes next semester or so we are going to deal with the cell biology class, separate class for 3 credit course. So I'm going to give an introduction for just 1 hour, just first lecture onto my cytology today. Okay? About the cell membrane and all those details. And tissues we will start different kind of next class onwards we are going to discuss. And then finally we are going to see a couple of organs, some very important organs in our system. Not all the organs, some of the organs. And then the next semester or so we will study the function of individual organ. That is called entirely physiology part. Okay? Physiology of these organs. That's comparative physiology or human physiology. Some of them you are taking now. That's good. That's the organ, that function of this organ. We call it physiology. Okay. And cell, the cytology, if you are going to study the immune cells, like T cells or B cells or the cells or white blood cells, how they are responsible for defending our system against any infection or disease, that is called immunology. That part, I think some of you are taking that class in this semester, like online, right? So that's only cells and how the cells behave to protect us. So this, it's at a different, it's ocean of knowledge on that particular cytology itself, or cell biology. It is there one. Okay. If you have no questions, I'll go to the next one. Okay, here identify different organelles in histological sections that we will see while, and then understand the functional roles for each organelle in cellular metabolism. We are going to study today this one. Okay. Cell, as I mentioned, you know about the cytoplasm. What is cytoplasm? Is a soup. How many different types of soups you have tasted in your life? Egg noodle soup, egg soup, bean soup, tomato soup, Campbell soup, right? It's ready-made. And Chinese restaurant, you got Chinese soup, different. And then English, that they serve a different type of soup on there, right? So traditionally, culturally, it may vary, different type of soup, right? So what is the variation from one to the other?
Vegetables, whether it's a
Right.
vegetable base or a meat base.
Correct. Right. We are talking about the cell here. What the cell soup, I told you about the cytoplasm. The cytoplasm of individual cell may vary. Like a soup, you may have large amount of protein, some of them are more carbohydrate, some of them are specialized functions in that. So that may determine, that cytoplasm will determine the type of cells, the component of the cells, because the nucleus of that particular cell will secrete a special kind of protein or increased amount of protein, or a type of a lipid is more. For example, in adipose cell or fat cell, what type of soup is there? They suppress all the protein, but their responsibility is to store only the fatty acids and triglycerides. Okay, that's on the fat. It's a fat storage. So that's the kind of stuff. So some of the cell will do the function for the immune system to protect our body. Some of the cells will store certain type of compounds inside the system, inside the cell. So that kind of stuff we are going to see in this class about colloidal gel surrounded by plasma membrane, like a soup. That's called cytoplasm. Mitochondria we are going to see, and you might have studied about Golgi apparatus and lysosome. What are those functions of this? We are studying here. Ribosomes, endoplasmic reticulum, and nucleus and nucleoplasm, DNA, and the chromatin chromosome. So we are covering all these topics in this class today. Is fair enough? Now, we will go on to the plasmalemma. Plasmalemma is the membrane. Okay? It's a 8 to 10 nanometer thickness. And what is that one? Thickness. Suppose I'm just, this is your cell, right? The nucleus. If you cut this part, and then if I elaborate that one, the cell, this is a nucleus, and this is the membrane. So the plasma membrane is that we call it as a plasmalemma, or the membrane which is covering onto the cytoplasm. The plasma membrane, otherwise we also call it as a plasmalemma as well. So we will see that plasmalemma is the one. It goes like this. Okay, this thickness I just draw here in a large way, like 8 to 10 nanometer. You know what is the millimeter? What is millimeter? Centimeter? 100 centimeter? How much? 100 centimeter is equal to 1 meter? Okay. So this is 10 times 100. 1 centimeter is equal to 10 millimeter, right? So just like mathematics, don't worry about this. 1000 centimeter is 1 millimeter. Then if you go further down, okay, if you have a micrometer, micron, the millimeter thinner, okay, 1000 micrometer, 1 millimeter, and then 1000 nanometer, 1000 nanometer, that is 1 micrometer. Okay, 1000 nanometer, 1 micrometer, 1000 micrometer, 1 millimeter, and 1000 millimeter, right? That is going to 1 meter. So it goes like this. If you have 100 centimeter, that's going to be 1 meter. Otherwise, you also call it as a 10 millimeter is equal to 1 centimeter. So it's a 10 times higher, millimeter and centimeter. So now you can calculate how much the nanometer. This is the unit of or the width of this membrane. Okay, this membrane is made up of the molecules. What type of molecules is there? These molecules in the membrane. What is the function of this one? This membrane function. It protects. Okay, it protects. Okay, as from the outside to inside. Also, act like a barrier. Right, is a barrier. You call it as a dynamic or flexible barrier, and site of the transport of molecules. Right, site of transport. So if I say this one, and there are certain receptors, some molecules are sitting on the protein. These are the proteins, and they help the molecules from outside. This is the outside of the cell, and then go to the inside. This is the inside. So the membrane will act like a barrier which protects the cell. Also, it permits. It gives it asks, do you have the permit to go in? So it will select you. It will not allow everything will go in, but the selective components, selective molecules move across. So these are the proteins which is sitting on the membrane will judge the transport of this molecule from outside to inside. So inside you have cytosol and nucleus and everything is over here. Okay. What other functions over these membrane receptors? Just I'm writing this membrane, okay? These are the membrane. Presence of hormone receptors. Hormone. The hormone which is floating around in the bloodstream, and they may come in contact with this cells, and then the cell will have receptors. Not all the cells will respond to the hormone. The cells that have the hormone receptors only will allow something like you have to have a visa to get into some countries, right? You cannot go directly. You have to have proper passport and visa like that. And here some of the port entry, they are going to verify you have insulin, you have the insulin receptors, then it will allow. It will not allow all the molecules getting into the cells. These are specialized cells. Okay, not all. So it will check, and then it will allow the molecules get across. So the receptors will do the function. We will see later on. And also this membrane will do in the nerve impulses. As I mentioned before, we will see later, these are the neurons. Okay, neurons and this is the nucleus, and then you have axons here and here. But it receives the communications, and then what the function of the neurons, it will transmit the message, right, to the next one, so that it will make a network. When I say the transmit, which part of the cell will do this function? The transmission of message. Transmission. Exclusively the transmission of the message is done by the membrane. Done by the membrane, plasma membrane. I got a plasmalemma, which is going to be a covering on that cell. Okay, so it will do this function, transmission of message. From one cell to cell. In other words, cell to cell communication. No cell, cell number, no cell, n cell, I put the no cell, another no cell, okay, or a no cell to another cell. So either way, the cell of the neuron onto the another neuron, cell to cell communication, or ordinary cell like a liver cell. Okay, here nucleus and this another nucleus. Okay, this cell and this cell will communicate through this membrane. So cell to cell communication in general, cell to cell communication, it will attach, this cell is attached to this cell, right? And this is being junction, there are some type of proteins, and it will say there is some toxic molecules are getting into the cell. Probably this is the 50th cell, and this is cell number 1. So you have several cells in the course of tissue. So this for the 50th cell will receive the communication of one toxin, and then it will alert the rest of the cells by communicating to cell communications with a messenger molecule or the signaling molecules from 50th cell to the cell number 1. So this communication, which is mainly by the cell membrane. So we will see how these junctions will do the message communication, or what type of signal they are communicating with each other. So that's very interesting. So the membrane plays a major role of exchange of information. Okay. Now let us see on the plasma membrane. I mean the plasmalemma. This is another the same thing. Plasmalemma or plasma membrane, one and the same, but in some cells they call it as a plasmalemma, where depending upon the function, but pretty much same. So when I say onto this plasma membrane, you cannot see under the ordinary microscope the structure of these cells. Okay, the structure will be seen by TEM. What is TEM here? Right, transmission electron microscope. So just I split this word like ideally electron microscope is in one word really, but I'm just going TEM. Okay, TEM, transmission electron microscopy. So using that, we can see the membrane, biological membrane. Okay, and they have got a Singer and Nicholson fluid mosaic model. Singer and Nicholson. Fluid mosaic. Okay, fluid mosaic model. Here. Okay, Singer and Nicholson mosaic model. This model is something like a membrane. Is a suppose I just want to use a little bit of animation. Suppose hold this one like a paper, right? So if I just circle this paper, and that is like a this like a cylindrical cells. You follow like this one, like the cell, right? So this paper sheet is a membrane, and then inside this you have there's a nucleus and cytoplasm, everything. So if you see the sheet of paper, just like a thin membrane, and then this is a surface of this membrane is a fluid mosaic. It's always mobile or motile or moving molecules. It is not like a stationary in a matter of fact, and always it's mobile. These molecules are moving ones to the other and do that. What type of molecules they are making on this? Let us see on to, if I draw of the cell like this and nucleus, and this is the membrane. If I cut this membrane and then it's elongate and under the transmission electron microscope, the lipid bilayer membrane. This one lipid and then another lipid and another lipid and another lipid. This is another lipid. I will explain to you what is this one, okay? Another lipid like that. So this called a bilayer. This is one layer of lipids. This is another layer of lipids. So lipid bilayer membrane. So that's why I want to get here onto this computer. Could you see this computer? Yes. Okay. Lipid bilayer that impregnated with proteins. That's why I want to get into your attention. So if you see the membrane, we are looking onto the cell, and this membrane is made up of lipid bilayer. What is this lipid? I'm going to little bit further. Then I'll explain to you. How many of you do you know about the lipid or fat? What is that? No? The lipid, whatever we call it, the lipid is a triglycerides. It's a glycerol. It's coming a derivative of, you know about glycerol? Glycerine. You know about glycerine, glycerol, and glycerine one and the same, glycerol. I'll draw a little bit of a cartoon like molecules. You just brush up your chemistry. CH2, CH and CH2. CH2OH, CHOH, and CH2OH. Carbon, how many valency for carbon? 4. So 2 hydrogen and 1 is carbon, another one is going over there. And then here 2 and 1 H and 1 OH. So these are the trihydroxy, otherwise we call trihydroxy ethane, right? Because the 3, right? 3 carbon, is that? Ethane or propane. Yeah, you are good. You are not sleeping. Thank you. Okay. 3 carbon, and here is the hydroxyl group, and this is called as a glycerol. I put it right. Glycerol, glycerol. If you say OL, all means alcohol. Okay, ethanol, propanol, all of them ends with OL, we got alcohol. The OL also correspond to the OH group. These are the trihydroxy propane, we call it as a glycerol or glycerin molecule. If this OH, if I add with an acid, suppose a fatty acid which I am adding to it, okay? So if I have a acid, I put it in a like a OH, and COH is an acid, organic acid, right? And then CH2, CH2, CH2 like that, you know, it's going on a long chain fatty acid, okay? CH2, like maybe 10 times, 10 and 14, so 14 carbon, and then finally you have CH3. It goes like this. I like this one. I just put over there CH3. Here you have a COOH, okay? And here I'm just in a short way, I'm writing this as a tricarboxylic acid. I mean, hydroxy group of propane, like a E, like this one, okay? So this one, OH, OH, OH. And this is COOH, right? And here the HOOC, HOOC, HOOC. So this is, I put R1, R2, R3, just to denote that there are 3 fatty acids, and the 3 fatty acids join with 1 molecule of glycerol. Am I correct? Okay. So if you do a reaction in a chemical reaction, biochemical reaction, the water molecule will go out. OH, the H from the glycerol, and OH from the acid will go as water. So another molecule of water, another molecule of water. So all this condensation or the reaction of OH or hydroxyl group and an acid group, they react together and to form 3 molecules of water. So the reaction, I'm going to write it here in a large way. This will go O, 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 and then you have a linking of all these are the linkers like a CH2 molecule, CH2 to the power 15 or 14, R1, and then this R1 is called alkyl group, right? R2, maybe this is a CH2, all of them are CH2 to the power maybe 16 or 17. This will go on R3 or CH2 to the power maybe 20 CH2 group. So if you see this glycerol is being esterified, we call it an ester, like acid and alcohol, a reaction, we call it an ester. I am explaining organic chemistry. Those who have done organic chemistry, you know very well. If you don't know the organic chemistry, please follow this one. This is one molecule of glycerol, but 3 molecules of fatty acids. These are fatty acids. 1 molecule of glycerol. 1 molecule of glycerol. And the elimination of 3 molecules of water, you will find an ester. So this is called 1 glycerol. Because of the ester, we call it as a glyceride. If you have 1 molecule of fatty acid, we call it of monoglyceride. If you have, and rest of them are OH, OH group. Like I'll explain in a different way now. Suppose if I write here O, and this is being esterified with R1, and this is OH and OH. This is called as a monoglyceride. GLYCERIDE. Okay, monoglyceride. So one word, monoglyceride. Suppose if the 2 of the hydroxy group were esterified with another fatty acid, this is 1 fatty acid, 1 glycerol, monoglyceride. Okay, another one, I write it here, 2, and this is OH. If you have 2 molecule of fatty acid and 1 molecule of glycerol is esterified, and this is called as a diglyceride because of 2 in a Latin word. 2 means diglyceride. Okay. And if you have 3 molecules, what will happen? 3. R1, R2, R3. So you call it as triglycerides. Okay? Triglycerides. So if you have these molecules in a way, if you have have you ever tested in your blood profile of triglycerides in your blood? Cholesterol and triglycerides. Have you done? Yes, Victoria? Have you ever tested? No, not yet. Okay. This is one of the culprit. If you have high triglycerides, it's difficult to get rid of. So you have to take some statins and reduce this formation of the esterification process. When you get triglyceride high concentration, whenever you are getting more of carbohydrates, the excess of carbohydrates that leads to more of glycerol, and this glycerol should be removed from our body. What will happen, or whatever the oil or fat substance you are taking it out, that will get into the esterification process, the triglyceride, and this triglyceride eventually stored in the adipose cells. And also this has been used, the diglyceride, especially not triglyceride, the diglycerides is responsible for making the membrane components. Okay, how that will make? If I write like this as a hydrophilic nature, and this is a hydrophobic. If I draw like this, R1 and R2. The same thing, but I am writing in a short form. Okay, so here this is the 1 molecule of diglyceride, and what is this one? This one is a hydroxyl group, because of the glycerol you have 2 component which is coming as a long chain fatty acid. The another one component under OH or hydroxyl group, which is ready to bind with the hydroxy moiety. And so it is something like if you put oil and water, it won't go mix it together. It's oil and water, just is a floating. The same way, if you think this way, the triglyceride, it is a hydrophilic nature. That means hydro means water and water loving. So water loving molecule is hydrophilic molecule, OH of that group. And this is a hydrophobic interaction because of the long chain fatty acid, which is not water loving but water hating, so it will go away from that. So that's why we make this this molecule like one is the hydrophilic, another one is hydrophobic region. So if you get one molecule, another molecule, and another molecule, and another molecule of diglyceride, another molecule of diglyceride, another molecule of diglyceride. So if you make this is form a one single layer of membrane. Suppose if you have another layer which is going along with this one here, this is constitute your lipid. It's made up of lipid because the fatty acids and glyceride, so called lipid. But there are two layers. So this is one layer, this is another layer. So we call it as a lipid bilayer membrane. Still we are in membrane. We are not even get into the cell. Okay, we have to study about the biomembrane. It's very important in the especially in the histology, because we are going to deal much on these conservations. So next one, what this lipid bilayer membrane will do. What is the function? The next one. If you see on to the next, this slide, if you see the peripheral proteins, because this lipid bilayer, I explained all this, so far my explanation for the lipid bilayer. If you say lipid bilayer, but membrane is made up of lipid bilayer. Everybody will say the same thing, but you have to understand the basic on that one. What is bilayer, which is made up of? And then if you have the impregnated with the proteins, what is about proteins? Peripheral proteins and transmembrane proteins, and we will see that one. What is that proteins over there? Suppose if this one lipid bilayer, we just put a one line like this, okay, and this is the green line which is going on the lipid bilayer on that, okay. And I'm going to write like a different color with this globular protein. These are the membrane protein. This is another protein. Okay. So these are the protein which is present in the periphery of the membrane. Okay. This is the one membrane. Okay. This is one membrane. This is the upper layer. This is the outside of the cell. Outside of the cell. So the membrane, and this is the membrane protein. Some proteins are transverse from layer 1 to here. Some proteins will go from here and then it will protrude outside. So these proteins, we call it as a transmembrane protein. These are the peripheral protein, these are periphery protein, and these are the transmembrane protein. Transmembrane protein. Okay. So they are present in the membrane, the biological membrane. So that's how you should understand that. What is the function of this periphery protein? They mainly as the receptors, these peripheral proteins. And some of the proteins are transmembrane proteins, and these proteins will act like communicating from outside into the inside, from outside to the inside, transporting molecules. Some of the molecules comes forming a channel or a small pore, and then it will allow the molecule go in without any hindrance from that lipid layer. It will allow, it will open a channel, so it will go through it. Okay. Apart from this, they also have glycocalyx. The glycocalyx is something like a carbohydrate moiety, which is present on top of this. I'm writing a different color pencil. And this is the red one, which is like a glycocalyx, which most of the cell membrane, they have it. This one. This one, call it as a glycocalyx. C-A-L-Y-X. One word, glycocalyx, which is present at the top. And it will follow a synchronous structure. For example, the glycocalyx is the component for the immune system to recognize one cell to the other, depending upon the carbohydrate moiety which is present over there. And then it will also get into the cholesterol. The membrane, they have got some other component like a cholesterol is also present over here. And the fatty acids, these are the cholesterol. So cholesterol is also present in the membrane. Protein, cholesterol, glycocalyx, and glycerides and fatty acid, all of them constituting the membrane proteins. In the structure of plasma membrane. Do you follow about this one so far? I told you the function is the transporter, and then across, and also the structure of the protein, as well as the function of this plasma membrane. Okay, I'll give you a little break now. You come back another 10 to 15 minutes, then we will follow the rest of the lecture. Okay? Okay, let us go on to the next part. We just to finish maybe 3 or 4 slides, okay? But we have to do 50 more slides to go, okay? Plasma membrane, we did study about the functions of that. Then one more topic I want to explain to you. As I mentioned to you, it is a lipid bilayer, okay? We explained what is those. The lipid we did with fatty acids and esterified with glycerol, one molecule. 3 fatty acid and 1 glycerol molecule. It will produce 1 molecule of triglyceride. Triglyceride, Triglyceride, glyceride. 1 molecule of triglyceride. If you have a 2 fatty acid molecule, Fa means fatty acid, plus 1 glycerol, that produce 1 molecule of diglyceride. Not diglyceride. Glyceride. And this diglyceride is mainly constitute the lipid bilayer membrane. So that's what we did study. What is the fatty acid? Fatty acid is the organic acid, like I'm writing the one fatty acid structure, like a CH3, CH2, CH2, CH2. It's a bond, CH2 and CH2. And then it will go multiply, and then finally it will end with a CH2. This is a continuation of a bond, okay? And then CH2, CH2 like that, and then finally you got COOH. So you can calculate the CH2 probably 14 or 15 or 16. So here you have if I do CH2, 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 all CH2, this carbon, if you see the carbon, 2 hydrogen over here, 2 hydrogens, 2 hydrogens. So all of this carbon's valency is filled with or full with the hydrogen molecule. In other words, the carbon valency is saturated. Saturated, okay? All of them are saturated with hydrogen ion or hydrogen atom in this case, hydrogen ions, hydrogens, all hydrogens. So the fatty acid, this type of fatty acid, we call it as saturated fatty acids. Suppose if you have another version like a CH3, CH, double bond, CH, CH2, then CH like that, and then you have a double bond CH2, CH, and then a single bond CH2. So you have, instead of 4 valency, you have only 3 valence, 1 hydrogen, but another valency is being bonded. So this is still, if you add a hydrogen, it can join with that group. So this part, this one, we call it as because of a double bond or triple bond, in some cases, we call it as an unsaturated fatty acid, because this is not saturated, so it's called unsaturated fatty acids. So the lipid bilayer membrane, as I said, fatty acid means you can also get a saturated fatty acid or unsaturated fatty acids. When you take some food and other lipid or the fat substance, if you consume, people they recommend more of unsaturated fatty acid. If you take unsaturated fatty acid content on a lipid diet or anything, that's the healthy. Your omega 3 or omega 6, omega 12, omega 2. What is that omega? That constitute the carbon atom. They get alpha, beta, gamma, omega. Like if you start with the fatty acids, if you call it like this, okay, this is the alpha, beta, and gamma, and delta, and then omega. You can say omega. So this particular carbon, you have a double bond. So it is a monounsaturated fatty acid. If you have a two double bond, you can get two or a di. That's a two bonds. Okay, two unsaturated bonds over there. So that you have a polyunsaturated, monounsaturated, diunsaturated, or you can also call it as a polyunsaturated fatty acid. Polyunsaturated fatty acid, PUFA. So if you go in your grocery stores, when you buy something on the fat combination of those commodities, when you see that one, if you say unsaturated fat and fatty acids like monounsaturated and the polyunsaturated fatty acid content. Have you seen that one? Any other food? What is that? And doctors say, nutrition, everybody will say that one. Those who have got some organic chemistry knowledge can understand, hey, it is a dice. There's a bond. It's not like a mono or di. Is an unsaturated bond is there. So that's why they call it as unsaturated. So you should remember that. Why I'm saying this, the lipid bilayer membrane is made up of fatty acid, which I mentioned earlier. Either they can, depending upon your consumption, the membrane is made up of saturated fatty acids or unsaturated fatty acids. Okay, the lipid bilayer membrane. If your membrane is made full of saturated fatty acids, is lipid bilayer membrane. That membrane with cholesterol and saturated fatty acid, it is a rigid membrane. Rigid membrane. Okay. If you consume with a PUFA or polyunsaturated fatty acids, if you consume a lot of them, then your cellular membrane eventually getting like a flexible. In other words, it's more fluid in nature. Why? Because of the unsaturated bond, and it won't give any rigidity with cholesterol. But with the polyunsaturated fatty acid, it is more flexible. Not only on the biological membrane, you also get into the nuclear membrane, mitochondrial membrane, and the other membrane, and whatever the membrane which is there, which is made up of lipid bilayer membrane. They mainly with the unsaturated fatty acid as well as the saturated fatty acid. So if you consume a lot of things, the ratio of SFA, saturated fatty acid, and polyunsaturated fatty acid, this ratio will determine the fluidity of the biological membrane. Why I am saying this one now, the diet which you consume, that reflect onto the biological membrane, the constituents of the membrane. And eventually, we studied about the function of the membrane, transport, and then it also produced the ATP, and there are different functions we did study. Membrane conductivity, communication, cell-to-cell communication, all of them depending upon this membrane fluidity. So if you consume a lot of only saturated fatty acid, what will happen? One thing is obese, number one. The obese lead to diabetes, and diabetes lead to colon cancer or atherosclerosis, and ultimately go to cancer, and finally that person becomes sick ultimately. So if you consume only saturated fat, why? Saturated fatty acid getting into the membrane, and membrane will, all of them are getting into the track of rigidity, and that leads to disease on that. Instead, if you consume both in a proportion, eventually if you consume more of polyunsaturated fatty acid in your diet, you are healthy, because you have extra bond. If anything happens, that bond will, like a buffering system, that will give a combination of extra hydrogen ion from other resources. So it will balance, as well as it can saturate. So even you consume saturated fatty acids, we don't have that many enzyme machineries convert the saturated fatty acid into unsaturated fatty acids. In other words, the reverse process. If you consume more of polyunsaturated fatty acid in your system, you have some enzymes or any other resources, you can convert it into saturated fatty acid. So our body can make saturated fatty acid, whatever is necessary. So the diagnostic aspect, one of the thing is if you collect the membrane, biological membrane, and then study the characteristic, whether that particular tissue excess of the saturated or unsaturated fatty acids. The ratio you can determine, and then you can determine the health status on that part. Okay. So that you should remember if you see the fatty acids and lipid bilayer membrane. Okay. Yes.
When you say polyunsaturated, that means that it can be more than one double bond.
Yes. Correct.
So if it's only one double bond?
3 double bond, 4 double bond, many. But because you have a carbon skeleton, maybe it's going on C24, maximum C28. The consumption of diet and food and nuts and all the oil, whatever you are taking in. But if you consume only donut with full of saturated fatty acids there, so that's real bad. Instead, you can take a omega 3, omega 5, omega 6, and the fish and oil, those things, or sesame oil, or you have good sources of monounsaturated fatty acids, peanut. It's very simple, highly protein and more of monounsaturated fatty acids.
I have a question too.
Yes.
You said ATP, etc. depends on the fluid.
That is correct. The reason is, and that's a good question. The reason is the mitochondrial membrane. Okay, if you say, we are going to see the mitochondria in a little while. This is the mitochondria, and then you have a double layer of this mitochondria, and then you have a cristae which is coming over here. We will see that in a little bit. And this membrane is the outer membrane, and this inner membrane. The inner membrane, there are two membranes. This is one membrane, lipid bilayer. This is another membrane of lipid bilayer. So this is the outer membrane, and this is the inner membrane. Okay, this is the mitochondria. The inner membrane, they have got the ATPase enzyme. This ATPase enzyme eventually will produce ATP from ADP plus PI. With the ATPase enzyme will act on this compound, and then it will produce ATP molecule. That's the energy currency. This enzyme will act if you have more of unsaturated fatty acids, which will flip-flop. That's what the thing, which it will have more fluidity of the membrane. If you have more fluidity of the membrane, the ATPase activity is heightened, increase. If you don't have that unsaturated fatty acid, this is more of saturated fatty acid, the ATPase activity slows down. You follow? So that will determine in the mitochondria. As well as some other function of the membrane transport, it follows a very good example, really. Okay, with this I am finished with the membrane, and I am going a little bit into the cell. So we are, as soon as when you get entry into a city, you have to get a permit, right? Just like a fort. And now we know what is made up of plasma membrane, and now we are getting into that, and we got the next one is our endoplasmic reticulum. Okay, they are also called as a vesicles, hollow, flattened, and cisternae, and rough and smooth endoplasmic reticulum over there, and 6 to 7 nanometer, and RER, small r denotes as a ribosome, or the rough endoplasmic reticulum, and S, small s, and then capital ER, that S stands for smooth. Okay, without ribosomes and involved in the synthesis of steroids and degrading enzymes. Why I'm explaining this, you have the quiz in all these PowerPoints. Okay, so you should know what the rationale behind the questions. Simply study ABC. Okay, this is C or ED. That is not going to help in any way. If you study, you have to study the basic. Then you will understand anytime, anywhere. Okay. So endoplasmic reticulum, where do you find this endoplasmic reticulum? With the cell, so I'm drawing like that. That's so you have the nucleus, and it will go like this, okay, and something like this going over there. Okay, some of these endoplasmic reticulum, which is having a rough endoplasmic reticulum. I put the blue mark, which is a ribosomes is attached to it. Okay, here this ribosome is attached to it. And another one which is here. So something like a beads on a string which is going over there. So these are the ribosomes. So the ribosomes is the one, the side, we are going to see the ribosomes in separately, but now for the sake of endoplasmic reticulum, we will say the ribosomes attached to this one. This is like a long halo-like thing, and these are some pathways, like a canal. Okay, they are attached to the ribosomes. Ribosomes involved in protein synthesis, and they are called rough. So it is a rough endoplasmic reticulum. Some of the other molecules, like this one, these are the smooth endoplasmic reticulum, and the smooth endoplasmic reticulum is responsible for. This is responsible for smooth, S-E-R, smooth endoplasmic reticulum, and these are rough or the ribosomal attached to it, endoplasmic reticulum. Our E-R called endo, E-N-D-O, endoplasmic, P-L-A-S, plasmic, reticulum, R-E-T-I-C-U-L-U-M, okay, endoplasmic reticulum. So, the smooth endoplasmic reticulum is responsible for, what is the function of the smooth endoplasmic reticulum? As I mentioned earlier, it produces a synthesis of steroids, and also degrading enzymes, like it involves in a drug detoxification. Whatever the medications or any other chemicals, the smooth endoplasmic reticulum is responsible for the hydroxylating cytochrome P450 system with hydroxylate. You will study when you go further on toxicology class, where we are extensively using this smooth endoplasmic reticulum and the drug detoxification mechanism there. Okay, and the rough endoplasmic reticulum is responsible for protein synthesis inside the cell. Protein synthesis, because you have a nucleus, and the nucleus will produce after transcription the mRNA will come out. This mRNA will join together with polysomes or the ribosomes, and then ribosomes produce more of the proteins, and the proteins being synthesized in rough endoplasmic reticulum, and then that is being transported, by vesicles and then getting into the Golgi. We will study in this class all about how the cell architecture is doing that function. Okay. Now we'll go on to the next slide on ribosomes. Okay. What these ribosomes is made up of now? Ribosomes are small and 15 to 25 nanometer in diameter, involve protein synthesis, especially in translation. And it is made of, there are 3 RNA. It's a rRNA, tRNA. rRNA is ribosomal RNA. rRNA, it is made up of rRNA, is a ribosomal RNA. RNA stands for ribonucleic acid. And tRNA, otherwise we call it a transfer RNA or transfer RNA. Okay, and then you have mRNA, that's called the messenger RNA. Where this messenger RNA is coming from? It is a transcription product from DNA, and where the DNA is there, it is from the nucleus. And this whole lot, we call it as a ribosome, and this ribosome is attached to endoplasmic reticulum, and that is called the RER. So the nucleus, they contain the chromosomes, and chromosomes they have the DNA, and the DNA, whenever there is a message is there.
We can't see.
Oh, I'm sorry. I'm sorry. Good to remind me.
Thank
Sorry, Victoria and Sugarland. Sorry, Victoria and Cinco Ranch. I am writing the rRNA, ribosomal RNA, tRNA, transfer RNA, mRNA, messenger RNA. All these RNAs constitute as a ribosomes. They call it as a ribosome. Ribosome is an organelle. So you should also understand what is an organelle is that. I'm right, it's like a O-R-G-A-N-E-L, organelle. Organ is a big, but organelle which is present inside a cell, like a plasma membrane is an organelle. Ribosome is an organelle, and mitochondria is an organelle, and nucleus is an organelle. Centriole is an organelle. Golgi apparatus is an organelle. So, and microtubules, organelles. All of them are organelles. They're the component of cell, we call it organelles. So ribosome is an organelle, and then this ribosome is made up of rRNA, tRNA, mRNA. So the mechanism of the ribosome, the main function of the ribosome is to synthesize the protein. From where? From the messenger RNA. The messenger RNA is coming from DNA. So it is a combination of all RNAs, and then they constitute the function. That's why last year, 2009, a Nobel Prize for this guy from Britain, and he got it for how these RNAs are arranging in a way that will produce of the protein. It's a very big complicated process of X-ray diffraction of this protein. They have done that work, and from the Cambridge they have done a very interesting work on how these ribosomes has been organized. So it's the latest information which we got it now. Okay, so the nucleus separate, we will see that one, but now for this class for this lecture we are doing with the ribosomes in this one. So there are some different kind of ribosomes. Will I just go through the PowerPoint? These ribosomes have a two subunit, large subunit and smaller one, just like this. The small subunit, the large subunit. So this is the smaller one, and this is a large subunit. These are the ribosome. Okay, ribosome. This is a large subunit. This is a small subunit. Smaller, this larger one. Yes, all done. Thank you. Okay. The small subunit and large subunit. Okay. Free ribosomes, they have a cluster. They call it as a polysomes used in intracellular protein synthesis. So the ribosomes act as a cluster. Some of them are free ribosomes. They forms a cluster, just like this, a cluster. Okay, this is another ribosome, another ribosomes, another ribosomes, another ribosomes. So there's a small and large, small, large, small. So what happened? They form the protein synthesis, which is the mRNA, which is in between that, which is feeding into that one. Whenever they go in, this is the red one is the mRNA, messenger RNA, and constitute the protein synthesis. The message is being translated. These are the message which is translated into polypeptide from one. So here this first like this, and then you have a second like this, and then third. These are protein. The green color is a protein molecule, long chain protein after the protein synthesis. So it's coming up like this. So that is the main function of the polyribosomes, or we call it as a polysomes. Polyribosomes, because it combination of different ribosomes together, so the polyribosome or polysomes, they produce more. They are synthesizing the protein. This is the first, and then when the mRNA moves in this direction, another molecule is added, another molecule. So there are several molecules of amino acid is being added. This is like amino acid, amino acids, amino acids, amino acids. So this amino acid chain which is growing. So this is the synthesis from the protein synthesis. I put it like that. Polypeptide synthesis or protein synthesis. That is the main function of the ribosomes. Okay. and there are the kind of the ribosomes, which is free ribosomes, polyribosomes, this synthesis, which is present inside the cell. I'll put it like this, okay. Cell and the nucleus, and there are some ribosomes which is clustered together. Is a polysome. It will produce the protein, which is going only inside the cell. These are the inside cells, okay. And I also mentioned earlier the rough endoplasmic reticulum also will have a protein, a ribosome attached to it, right, which is going over there, right. So these ribosomes, which is present in the rough endoplasmic reticulum, that also produces the protein synthesis, and that will produce the protein and export outside the cell. Otherwise, we call it as a extracellular protein. See, some of the proteins we have got on our bloodstream. How do we get? It is coming from liver cell. The liver cell will produce using the rough endoplasmic reticulum and the export outside the cell, and that is called the protein, which is the extracellular protein, which is export outside, which is present in rough endoplasmic reticulum. And another type of cluster, we call it as a polysomes, which is not combined with the rough endoplasmic reticulum or the endoplasmic reticulum, but it is on separate. It is not attached to any of the rough endoplasmic or endoplasmic reticulum, but it is being floating inside the cytosol as a cluster, as a polysomes. We call it polysomes. They also produce the protein, and that is present inside the cell. It is not export outside. You have to understand that. There are in an industry, sometimes they have to synthesize their own product, keep it outside, export quality they call it. And some other, the same material, but they produce, it's only for internal, just only the national level they will give. So export quality is different from national quality. So they want to do it. But though they both are the same, the product, but they want to distinguish. Like this, the extracellular proteins that's been synthesized from the rough endoplasmic reticulum, they are called the polysomes, which I explained to you. Polysomes is an individual cluster. It is not combined with it's not with the rough endoplasmic reticulum. It is individualized. But it produced the protein present inside the cytosol, inside the cell, not exporting outside, and they are called as intracellular protein synthesis. Intracellular protein synthesis. Okay, intracellular protein synthesis. So one type of protein present inside the cell, and some other protein synthesized and export outside. Why? The rough endoplasmic reticulum, you have I told you, like a canal, a pathway. So as soon as the protein is synthesized here, it will export outside, so that it is compartmentalized. It is not mixing with the protein inside the cytosol. It synthesizes the protein outside. So a nucleus will producing the mRNA of a particular hormone, like a peptide hormone. If that is being synthesized and then in the rough endoplasmic reticulum, and that is going to be again exported outside. Now the hormone is being secreted outside. So this hormone is never ever be inside the cell, and it is not activating this process. So it's going to be exporting outside. Do you follow? Because if you say, yes, ribosomes involved in protein synthesis, now you have to go one step higher. One type of polysomes or ribosomes will produce more of protein that is present for the requirement of the cellular material. There are certain enzymes, there are other proteins, and the nuclear protein and the mitochondrial protein, all these proteins, they require the protein synthesis. That is from this polysome which is coming here. Some of the message which is from outside, hey, I need more of this protein, why don't you synthesize and give it to me? So export order. So as soon as the nucleus receives that information, it will produce more of mRNA that is being translated in the endoplasmic reticulum via, and then it is going to export outside the cell. So that is called extracellular protein or any other function. Do you follow that one? So ribosomes, if you say there are two types of function, is intracellular protein synthesis and extracellular protein synthesis. Yes? Okay, now we'll go on to our next slide. We call it as a vault, not valet, it's a vault. Okay. They are newly identified organelles of 35 to 60 nanometer. If it is unstained means different chemical reaction to see, visualize it. If it's unstained, it's like an octagonal in shape, and they are present near the nuclear pore. Suppose it is cell, okay, and there's a nucleus, and this is the nucleus, and these are some of this nucleus. They are the nuclear pore. Okay, I think yesterday we explained our histology lab the nuclear pore. That's a material where mRNA will go out of the nucleus through these pores. So, used in transport of chemical message between the nucleus and cytoplasm. This is something like a trafficking molecule. They are sitting, I put it in a different color. So, these are green, this is green color, which I am writing here. And they are floating near the nuclear membrane, and it will help to take some molecules which is present in the cytoplasm. These are the molecules. So, it will combine with this molecule with this one, and then it will transport across, and then from inside to outside. It will take like a carrier, like a courier, I can say that one. Take one, like a postman from one destination to the other. So it will carry the message from one to the other, on one molecule into the inside and outside. So that's what is used in transport of chemical message between nucleus and cytoplasm. So that's the function of vault, another newly identified organelle. You follow now? Vault, newly identified organelles. And now we are going on to the mitochondria. So if you say inside the cell, you have a membrane, nucleus, nuclear membranes, and rough endoplasmic reticulum, smooth endoplasmic reticulum, polysomes, and now got vault. We did study about this, and now we have a bit of mitochondria. So just I put the green dot like this. I'm going to explain later how this. What is the mitochondria? Under light microscope, appear thread-like thing under light microscope, but that's on the 0.2 to 1 micrometer. Okay. In electron microscope, it looks like a rod-like structure. Electron microscope, EM, and is an ovoid trilaminar membrane. What is that trilaminar membrane? You will see that one. Outer membrane, fatty acid synthetic enzymes. Then inner is folded cristae and ETC enzyme, you call it the electron transport enzymes. And there are binding sites for calcium ions and ATPase enzyme complex for aerobic oxidation, and they are the powerhouse of the cell. That's the energy currency of ATP is being produced by the mitochondria, right? So when you see the structure of this mitochondria, I will draw like this, which is one. This red is one membrane, outer membrane, and then I am drawing a second inner membrane. You see under the microscope, an electron microscope, and you will see the structure of this mitochondria clearly. Okay, so the red one, which is the outer membrane, the green was the inner membrane, but it is folded. Could you see that one, the folding on that one, inner membrane? And in between that, if you see the part, red one and then the green one, in between there's a gap. So there are proteins which is sitting in between that one. Okay. Proteins, P for proteins, which is there. Yeah. So if you see this one, 3 layers, so we call it a trilaminar. They call it trilaminar membrane. That's why we call it as a membrane. It's made of 3 layers of this. One layer, but each layer, as I mentioned, if I say one line, one membrane, means it is made up of lipid bilayer, right? Bilayer. This is also bilayer, and this is also bilayer. always remember that. That's what I explained to you in biomembrane, the first one. Membrane, if I say membrane is made up of two layers, outer layer, inner layer, lipid bilayer membrane. Imagine. So here, two layers, and here, two layers. And in between the layers, we have the proteins. What type of proteins are present inside of this mitochondria? We call it as an electron transport chain membrane, like a
Sorry.
TCA cycle membrane.
The inner membrane is also.
Cristae, they call it as a cristae, 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 inner membrane. And also they have the machinery, like a ATP synthesizing machinery. I put it the red line with this here. So what happened, this inner membrane is responsible for highly intact TCA cycle enzyme, tricarboxylic acid cycle, TCA cycle enzymes. And these TCA cycle enzymes, they produce more of NADH plus and H plus substrate for the electron transport chain, and then this will eventually get into the electron transport chain, will produce ATP. Like have you done in your biochemistry class TCA cycle, and TCA cycle will produce more of NADH and H plus. And if I write electron transport chain, it is made up of iron sulfur protein, FES, and there are different sites of the complex molecule, site 1 and site 2 and site 3, and all of them, these complex iron sulfur molecules from this, when they go through, the electrons are transferred to the one to the other, where the ATP is being produced from ADP and PI. Then ADP and PI. Okay. Another molecule of ADP and plus PI. Depending upon the substrate, if NADH is the substrate, you get 3 molecules of, it will go and feed, and if there are 3 molecules of ATP will be produced. ATP molecules produced for 1. And it goes through this electron. Where this is coming from? This is coming from a TCA cycle, tricarboxylic acid cycle. Where the TCA cycle is being operated? From glycolysis. Where the glycolytic pathways is coming from? That's from glucose, what you eat. And whatever you eat, the glucose or any of the protein, if you eat protein, more of protein, that will feed the amino acid to TCA cycle, and TCA cycle will produce. So whatever we are eating here, or if you have more of fatty acids, that's on the beta oxidation will produce more of NADH. It will feed to NADH, will produce more of NADH, or reducing equivalent, and that reducing equivalent will involve transfer of the electrons in electron transport chain, which is responsible for the ATP. Okay. This process is the bioenergetics, and that is exclusively for biochemistry. If you are doing biochemistry, you will know. But this entire process is present in this cristae, this membrane, in the mitochondrial membrane. Do you follow that, Paul? Where the mitochondria, what is the function of mitochondria? Where the electron transport chain, where the TCA cycle enzymes are there. That's why we call it as a powerhouse. So these molecules are, this mitochondria is floating inside the cell, right? These are the cell. And the cell will receive all these glucose molecules, and glucose, their glycolysis is taking place in the cytosol, and the TCA cycle present in the mitochondria, so it will go into the mitochondria will produce ATP, and then once the ATP is there, and then pumped out, this ATP out the cytosol. So this ATP is floating around, and that is changing the calcium level as well as import and export, whatever the requirement. It will be useful. That energy is being used for different purpose for the cell function. Like we have gasoline to run our car, or battery to run our car. For run our cells, we need more of the ATP, right? So the ATP is the power source from this. I normally call it that ATP synthesis, or the mitochondria is the powerhouse, or the energy house. The Houston is the energy center, right? So imagine, are we synthesizing more of ATP? Gasoline, right? Is a more of gasoline we are producing it. So that's the powerhouse we normally call it as. Okay, now we will go on to the next one. Golgi complex. this Golgi complex, we call it a Camillo Golgi. That's in 1898, more than 100 years old. Okay. And network of filaments near the nucleus and identified by silver or osmium ion. There is a typographical error. The L is missing here. So silver or osmium ion. So using that ion, we can identify the Golgi complex. Okay. All cells except the RBC and keratinized epithelial cells, and they are 6 nanometer thickness, and cavity is filled with secretory proteins by glycosylation and export to extracellular from endoplasmic reticulum. What I'll do, I'll stop in the Golgi complex. Next week we will start as per of the syllabus onto this class. Do you have any questions from Cinco Ranch? You follow? You got all those who came late, please go ahead and get the HTTP site where I have uploaded my syllabus as well as the PowerPoint. But eventually you will able to log on your Blackboard as a supplement. It is not an online, your face-to-face class. I expect you to be present in the class. The attendance is compulsory. Okay? So I am counting the head from Victoria, Cinco Ranch, and Sugarland.